project conference. This is the electrical and computer engineering section. Next project. These gentlemen will introduce themselves, so thanks all for coming.
Thank you all for coming, and we'll try not to hold you too long before lunch, because we know it's right after our presentation. I'm Justin Leek.
James Novorita.
Scorza.
And this is our presentation on short-read genomic sequencing using FPGAs. Getting into our overview, we start with DNA sequencing and how it works, the need for our design, the specifications, what an FPGA actually is, the tool sets we developed, the string searching algorithms used, the time complexity results, power efficiency results, bill of materials, and recommendations and conclusions. And now we'll talk about DNA sequencing.
Thanks, Justin. So with the DNA sequencing, just give you a little biology background. The DNA consists of 4 different bases. And with these bases, they fall into 2 categories, and there's 2 different pairs, C and T, and A and G are paired. So we are using next generation machine, using the parallel sequencing method. And with this method, the DNA enters into the machine, and then what the machine does is it can do millions and billions of reactions at a time. So that's why it's very useful. And it attached itself to, what we do is we attach it to a synthetic primer to the fragments of the DNA. And then what we're trying to do is figure out the sequence of those fragments and then get those results. So what happens is during the reactions, while it's trying to figure out what the bases are that come next in the fragment, different light emits, and then that tells us what the next base is. And then once we have all the DNA sequence for the short reads, those fragments, all that gets exported, and then our job would be to try to figure out what order those fragments then go into. So again, this is the overview of that process, and then I'll hand it off to James, and he'll talk about the short reads.
Thanks, Kevin. So, as Kevin mentioned before, there's something from the next generation sequencing machine that are spit out called short reads. These are sequences between 10 bases and 200 bases long, usually. And what we're trying to do is we're trying to find pieces of this that match to a reference genome. So in other words, if you give a blood sample, the idea is that we analyze we take your DNA out, and then we see, okay, the next generation sequencing machine says there's small pieces of this person's DNA that might prove important. So what we do is we take those and map those to something that might be able to be identified. For example, like a strand of HIV. There's 9 or so different strands, so each of those pieces could be important in identifying which one. So in computer science, you need to use what's called exact string searching, and that's what we did. And that's the most computationally intensive part of this entire DNA sequencing process. So we're trying to expedite that and improve some power. So it's kind of analogous to putting a puzzle together. If there's a small piece and it won't fit inside a bigger one, you got to keep searching through the lot. And moving on into some history about whole genome sequencing. So in 1977, the first whole genome was sequenced, and it was trying to be understood by hand. So it took 7 years to understand. It's called the Epstein-Barr virus. It's actually fairly small, too. So they thought, California Institute of Technology said there's got to be a better way. So they decided to make a semi-automatic sequencing machine that came in 1986, and then a fully automatic one came a year later. Some new technology some technology that's being used, or sequencing methods that are being used today, are things like 454 pyrosequencing is what the next generation machines usually use. And there's also new methods down in the lab being studied, such as mass spectrometry, Sanger sequencing, and also some other methods as well. So you're probably wondering what kind of impact whole genome sequencing has today. Well, it actually has a lot more than you think. So it's now a vital tool in the modern world of medical decision making. New technology has made this process easier and more affordable, so that means a larger group or context will be able to apply to it. So medical researchers use this stuff, as I mentioned, for HIV identification, but also they use it to understand rare genetic disorders and even try and combat cancer. So the foundation laid here is pretty large, and it's only on the up and up. So the future could mean that one blood test could, in hours later, you could get your results of what could potentially happen to you in the future, or even just what needs to be done with you. So Justin's going to talk a bit about where we fall in this process and how it's done.
So the need for this design would be to utilize the FPGA to optimize the string searching process due to its time constraints. And the faster this can be done, the faster patients will endure less suffering and pain. Getting into our specifications broken into 3 parts, starting with the physical. We are required to use the Intel Cyclone 5, which is on right here, and which also includes a A9 embedded microcontroller. We're required to use the i7 CPU host that's down in the lab, and that host had to have 24 gigs of RAM, allowing us to compile an OpenCL. Functional requirements, we're required to use OpenCL, allowing the use of C for ease of access for us. We also had to compile a Hello World program as a start. And we also had to use basic string searching methods. And the 2 we chose were the naive and Boyd Moore. And in doing so, we had to use matrices and nested loops to optimize for spatial locality. The ethical standards, we had to follow the standard for analyzing raw data from IEEE. And since this is a medical field project, we also have to follow the HIPAA standard. Getting into the hardware a bit, this is the Intel D1 SoC FPGA. Well, that's the whole board, but this chip right here, that's the FPGA, as well as the A9. And it's OpenCL compatible, and it's an optimal for our application because they can get pretty pricey, as I'll explain later. Getting into what an FPGA actually is. It's an IC, it's a reprogrammable IC where routines can be divided into parallel fashion, much like with the parallel circuit. So they're independent of each other. And FPGAs also use hardware descriptive languages, such as Verilog or VHDL, to interpret how to configure logic circuits. And now we're getting to why an FPGA for DNA sequencing.
Yeah, so this is a good question, right? We talked a little bit about DNA sequencing, and now FPGAs are being shoved in. So let's break it down a bit. So DNA analysis, as said before, needs to have a lot of things happen at the same time. So the problem is most modern computers run von Neumann architecture. And I'll discuss this a little bit in a little bit with a diagram. But that means that processes run concurrently. So to you, it seems like things run at the same time, but on the low level, that's not the case. In order to run 2 things at the same time, one needs to go and perform an operation. It needs to stop, sync up with the clock with the second one, and the second one needs to operate, and they alternate back and forth. The reason this seems synonymous to you is because the time delays are so small, but the board sees it as downtime and uptime. So with FPGAs, you don't have that problem since, as Justin said, it's much like a parallel circuit. Things are done separately, so you don't get that weird architecture on the FPGA. So with FPGA, specific circuits can also be tasked to do certain things to make things faster and more power efficient. So for DNA sequencing, you can have multiple circuits built to work on one task, and with OpenCL that's nice because it allows for workgroup theory. You can compile certain pieces of code to run the FPGA on certain tasks, and this overall, as I said, will lead to better power efficiency. So as I said before, with von Neumann architecture, you have a typical fetch, decode, and operation cycle. So imagine this blue line here is Google Chrome, and yellow is like iTunes or something like that. So you have a fetch cycle, and in order for that to keep going and have something else run with it, another iTunes needs to fetch its instructions as well. Then they need to decode and operate. So these time delays are small, but it's kind of gymnastics to get the circuits to work, and it overall uses more clock cycles. So that can be more programming, or that can be more power intensive. Whereas an FPGA is a bunch of logic circuits that you build through programming syntax like Verilog and VHDL to perform certain tasks. So there's no force fitting for this process. So getting into the software that we use OpenCL's 16.0 from Altera, this FPGA SDK. We use the C programming language, and in order for Altera's OpenCL to run, we needed Windows 7 64-bit. So here's the cool thing about OpenCL. This is a new platform that allows for FPGAs to compile and run C code, but in order for that to happen, this needs to, the Altera OpenCL compiler needs to compile that code and translate it into Verilog or VHDL. And then it's a binary file that executes on the SoC. Now the great thing is that a lot of FPGAs now use PCI Express, so that's a bottleneck when you're talking about data transaction. With this, all the pins are connected together, so they just operate and transmit a lot faster. And that's what the host program does, it kind of data management. It uses data management and wardening to make things faster and more power efficient overall. Now I'm going to hand it off to Kevin. He's going to talk about how this fits into DNA sequencing.
Thank you. So when you get the information from the sequencing machine, that's basically considered raw data, and that's what we're starting up here, the genome raw data. And then what we need to do is process this. So we're going to write code in OpenCL, and that will compile into Verilog, and then it will run in a main file and a kernel file. So basically, the kernel file does the heavy lifting of doing the operations, and then the main file does the data management. So once it's done, it gives us a result, which will be the placement along the target DNA that those fragments should be assigned. So when we started the project out, we had to do coding two different ways. We had to do it on the kernel and on the host. Then we had to compile it on both, transfer and execute on the FPGA. A little cumbersome, and we did develop a tool set to help make that a little simpler. We'll go over in a moment. This is an illustration of the screenshot of the kernel design. So basically what we're showing here is we are writing this in C, and we are also doing nested loops here to help with our indexing and optimize our spatial locality. So the tools that we did develop, we were able to go online and find a DNA generator, and what we do is we just enter the number of bases that we're looking for. I just put 1,000 in here for the picture. Obviously we were using 100 million, so it'd be a little more than that. But then it gives us a sequence of DNA. So what we did is we took that DNA sequence, put it into an Excel file, and then it automatically broke it up into fragments. And we did it two different ways. We have one of standard lengths and one with random lengths. So we were able to use that for our testing experiment. And then the second tool set that was developed, James was able to write a program to help with the previous slide I was talking about how cumbersome it was. We had to use 4 different terminals, and he was able to make it easier, and with us just using one terminal instead of 4. And the neat thing about that is we were able to upload that online, and other people are using that, downloading it and using it for their developments. So now we'll hop into the string searching algorithms. So this, again, is the most time-consuming portion of the DNA sequencing. So it's important for us to optimize that. That's what our design focused on. So we use 2 different algorithms, naive, which is also brute force, and the Boyer-Moore. Naive is more of a linear design with the length of the genome and the time it takes to find the match. And then the Boyer-Moore is more sublinear. So for the naive approach, basically what we're doing is we're taking the fragment and we're looking at the first position and comparing it to the first position of the target DNA to see if those are a match. If they are, it continues down to the next positions until it completes and finds a match or not. And then it simply moves the fragment over one spot and then rechecks it. And it does that through the whole 100 million bases. So it's very time consuming. This is our theoretical graph. So basically for this, our time complexity is big O, n times m. N being the length of the genome, m being the length of the target DNA. And when we processed 10 to 100 million bases, and our results showed just that, a linear relationship here. And time complexity is not the best for this method, so you're probably thinking there's got to be a better way, and there is. And I'll pass it back to James, and he'll talk about that.
Yes, so Boyer-Moore, this is a combinatorial approach. That means there's multiple rules of the algorithm. The first one is called the good suffix rule. And the good suffix rule searches through the target DNA right to left and finds pieces of it in which that are a match to the sequence you're looking for. And then it shifts and aligns into an array. Whereas the bad character heuristic does the opposite. It finds where the places are mismatches. And the reason this is so much faster than the brute force implementation is because it utilizes something called pre-processing. Pre-processing is upon compile time, it figures out exactly where the mismatches are inside of the target DNA, so it knows on runtime, oh, I can skip these places. So this is a lot faster. As you can see, the best case time complexity is O to the m, which is just the length of the sequence. That's only if there's one match. But there's also the practical example has been found in many papers to be O to the 3 times n. Now, here's our results. As you can see, it's a lot faster. At 100 million bases, we're talking about 7 seconds versus about 710. So it's sublinear as expected. Even though this is only a partial implementation on the FPGA due to time constraints and a few weird things with the pre-processing directive, OpenCL takes about anywhere from 2 hours to a couple days to compile from C to VHDL. So we would have to compile it over and over again to run these tests, and we didn't have that kind of time. But the improved speed efficiency, the speed up ratio was 110 regardless of the partial implementation. We're double E's, we care a little bit about power. So let's hand it to Justin. We'll talk about that.
All right. From this graph, you see that we compared the FPGA and the A9 chip, and right here we saw about a 1 watt difference in focusing on the naive. And you see that the FPGA slightly outperforms the A9. And getting into the Boyer-Moore versus the naive, you see that this blue graph is the Boyer-Moore, which uses more instantaneous power, although average power would be less. And the bill of materials, which was the Cyclone 5 SoC, we split that between us, D-Reyes, and that was, again, the optimal choice because some of these can get to $30,000. Getting into our specification completion, we hit all of our required specs. Now we'll get into more of the conclusion.
Back to the naive, we were able to fully implement the naive on the FPGA. So this graph here shows the difference between running it on the host computer and the FPGA. So with the shorter lengths of the genome, the speed up ratio was a lot faster. However, when you increase the genome length, it was negligible. It was 1.29 of speed up ratio. So not a very efficient way of doing it. But on the next slide, it shows also the power that was consumed during the naive process. The smooth lines on the on the graph indicate that we had gathered a lot of information and ran our results several times, and it was pretty consistent. And that's why we get the smooth surfaces. So, and the power, as far as the genome length increased, the power consumption also increased, which would be expected.
Right, and for the Boyer-Moore, this was a pretty dramatic speed up from the naive, as we said before. We had to use the log scale to even indicate some difference because this graph is unreadable. So you can see that there's just an enormous, this is a true superlinear versus sublinear relationship. So at 100 million bases, we saw a speed up of 110. This really becomes more evident in the differences of the naive. And as we look at the power, as Justin said, the total power consumption is less because even though the Boyer-Moore was 200 milliwatts larger as far as power consumption goes, it was also 110 times faster. So the total power consumption is also less. Another thing too that's pretty cool is this Boyer-Moore implementation has never been done, according to our research in academic journals, in OpenCL. This is the first time anything like this has been done. So this is something that's interesting to us. And Justin's going to finish up here.
So the big conclusion of all of this that we found is OpenCL on the FPGA is useful for speeding up development without going directly into HDLs. And we also saw that the FPGA performed on a similar level as the embedded host we use, as well as it consumes slightly less power as the host we use. And we see that in the long run, FPGAs will be useful for saving power consumption on a larger scale. So think in some sort of lab where you have hundreds of these FPGAs running at the same time, and for that lab, it will save lots of power. Also, recommendations for a further continuation of this project would be to use the Boyer-Moore on the FPGA a little more. We only partially implemented it, as well as the next project could do OpenCL versus VHDL or Verilog direct approach. And we see that an FPGA could be put directly into a sequencer to lead to much faster real-time results. And that's the synopsis of the project.
And our group would also like to thank Dr. Fahad Saeed for his knowledge and guidance on this project, as well as his hospitality in the PCDS lab. And we'd also specifically like to thank Usman Tariq, a master's student here. He helped us a lot with OpenCL for FPGA, as he's the resident expert. And at this time, the group would like to open the floor to questions. Thank you.
Everybody speak up. Questions? Yes.
And nice job, guys. With the OpenCL, so I've done FPGA stuff in PHDL, but not OpenCL. How do you control what stuff runs in parallel when you do OpenCL, when you're essentially writing C?
That's a great question. Thank you. So that is done with kernel. So as stated before, kernels are written. OpenCL calls the C compiler that translates to VHDL a kernel. These kernels have what's called workgroup theory in OpenCL, and they have directives of how to get them to work together. So what you do is you set up scoped buffers that you can pass through to OpenCL that work together, that cross each other, and you focus those specific transactions on one particular part. So for example, like let's say one loop you have going through to search the DNA, and the other one you're keeping track of what's going in the array of the match. Well, you can have one kernel working on that specific loop outside, and you can feed it into the scoped array of the second loop. So that means that they're working together, but that OpenCL's job is to kind of take that and make it less complex for people who don't know, for say, VHDL. But the problem with that, the trade-off, is that VHDL and Verilog is faster if you really focus on it, because you're following OpenCL's compiler directives as to how they optimize the hardware. So it's not a perfect solution, but it does have some benefits, at least to getting more power efficiency out of the FPGA.
So it really comes down to how you divide up your kernels. So
Exactly.
each of those kernels kind of run like their own standalone C program.
Mm-hmm. Yes,
and
exactly.
then you guys have to break it up to get parallel.
Exactly. It's like passing variables through functions in C. Does that answer your question?
Yep, no.
Cool. Any more?
I have a question.
Sure.
What is your outlook or projection for taking this, the OpenCL technique versus the Von Neumann centric technique, and how would that project out into, we'll say, DNA analysis in, for let's say patient DNA, or what would the speed up be or the effects be? That can you speak to that from your results here?
So with the FPGAs, you're able to get real-time results. So it's as James was explaining with Von Neumann, it doesn't run through parallel. So the speed-up results were most efficient with the Boyer-Moore method. We weren't able to fully implement that method, but with what we did, we were able to achieve quite a bit of speed-up.
110.
Yeah, 110 times the speed-up. So is that answering your question?
So you would expect to see like 110x improvement over like conventional method, just using a pure software approach?
There's actually potential to be, I wouldn't say more, but yeah, because what you could do is you could put the FPGA inside of the next generation sequencing machine, and as data is funneling into the FPGA, it processes it while the genome data is still coming in from the sample. So you can actually probably do it a lot faster. And that's what Dr. Saeed was talking to us about just recently. He said that that's kind of an idea that's being pushed around in the community now.
Okay. So if you implemented the full extent of the Boyer-Moore algorithm, would you expect that processing improvement factor to go up?
Yes.
Yep.
Okay. And do you have any feel for where the boundary might be on that? Would it be 200x, 500x, 1000x?
We really haven't.
No, we don't have a limit, but I know we aren't, with OpenCL, we aren't able to fully implement Boyer-Moore, in that the pre-processing has to be done at compiling time. So that's one disadvantage with OpenCL. But if we were to use the Verilog or to write our coding, then we could manage the hardware, put out pre-processing, have that done at the same time, so we have 100% of it running on the FPGA.
Mm-hmm.
So then that would even increase further.
Okay. Thanks.
More questions? If no more questions, let's thank this group. There will be a break for lunch, and we have one more group that will be presenting at 1 o'clock today. So if you're interested, come back.